Okay, so the first topic of the video is glycolysis. Since this is a 15-week supplement course, the best way for you to really absorb this course is to do it with our other 15-week program. So there is a pretty in-depth lecture that I actually did for our last 15-week course in glycolysis. So I'm just going to go over it briefly here because just keep in mind these supplement videos, you should be watching them in conjunction with our last 15-week course as well. So a quick intro to cellular respiration, excuse me. Humans oxidize glucose from start to finish through glycolysis, the Krebs cycle, and the electron transport chain. So those are the three processes in the human body that will generate ATP. In the first two steps, not much ATP is produced directly. Glycolysis produces 2 ATP per glucose molecule, and the Krebs cycle produces 2 ATP per glucose molecule. So out of the three processes, those first two aren't great for producing ATP, but they do produce a lot of other things that are important for the ETC. So as glucose is oxidized throughout these processes, the electron carriers NAD+ and FAD are reduced to their high energy forms NADH and FADH2. So their high energy forms, they're also referred to as reduced forms, and their job is just to carry energy throughout the different systems in the form of electrons. So NADH and FADH2 will later be used to supply electrons to the ETC to develop more ATP. So glycolysis also produces 2 NADH molecules per glucose as well. And per each glucose molecule, which actually results in 2 spins of the Krebs cycle, because as we'll go over really quickly, once you get to the Krebs cycle, it's actually 2 3-carbon molecules, so we can spin it twice, and we're going to produce 6 NADH and 2 FADH2 molecules. So these products feed into the electron transport chain where 34 ATP are produced per original glucose molecule. So it's a lot more efficient in making ATP than the first two, than the Krebs cycle and glycolysis. All right, so we'll start with a quick overview of glycolysis, and then we're just going to go through the steps pretty quickly. So glycolysis is highly conserved throughout all domains of life. Any type of life that you're going to look at, they're going to perform glycolysis in the cytoplasm. It's the most highly conserved. It's one of the oldest processes. It's just the way, that we can make energy. So in fact, all living cells contain enzymes for the glycolytic pathway. So it's split into 10 steps, and these 10 steps, you can subdivide them into 2 different phases. So the first 5 steps are known as the preparatory phase, and the last 5 are known as the payoff phase, just because the flow of energy is working in each of those phases. So in the preparatory phase, we're actually spending a little bit of energy for a bigger payoff during that second half payoff phase. So also keep in mind that glycolysis takes place in the cytoplasm. And with both of those in mind, we'll just go through the steps. So during step 1, a singular glucose molecule, that's our starting molecule, is going to enter the glycolytic pathway when it is phosphorylated into G6P by hexokinase. That just means that we put a phosphate group right onto that 6-carbon of the glucose molecule, and the enzyme that is responsible for doing that is hexokinase. So G6P and hexokinase work on a negative feedback loop. An increase in G6P, which is the product, is going to inhibit the activity of hexokinase. So we start making a lot of product, the enzyme is going to be inhibited. The product starts to get a little bit lower, the enzyme is going to get more active to create more of the product. G6P is now unable to leave the cell because of the negatively charged phosphate. So that's really the main point of adding a phosphate onto the glucose, is that it sequesters it inside the cell and it's not able to pass through. So G6P is also supplied to the glycolytic pathway by cleaving glycogen, and step 1 is irreversible, so that's considered one of the committed steps to glycolysis. So as soon as a glucose molecule is phosphorylated with that phosphate group on the 6th carbon, it's pretty much committed to going through glycolysis. Okay, so step 2, G6P is converted into F6P, and the name of the enzyme is phosphoglucose isomerase. And this step converts the molecule into a usable form for the following steps. So we're just taking that 6-carbon sugar, that glucose, and we're turning it into fructose. So another phosphate group is added to the F6P molecule via the enzyme phosphofructokinase 1. It's more commonly referred to as PFK1, and it produces a molecule called fructose 1,6-bisphosphate. So like this step, like step 1, is also irreversible. So this would be considered the second committed step of the 10-step process. And it actually uses up 1 ATP molecule in order to turn in order for PFK1 to actually operate and produce fructose 1,6-bisphosphate. So this is referred to as the actual committed step of the entire process because the molecule can no longer be used in any other metabolic pathways like glycogenesis. So when we're at step 1 and we convert into G6P, there actually are a few other sort of different processes that G6P can undergo besides glycolysis, including the phosphate pentose, the pentose phosphate pathway, which we're going to talk about in a little bit as well. But once we get to step 3 and we convert to fructose 1,6-bisphosphate, there's nothing else that this molecule can do except finish glycolysis. So that's why we refer to step 3 as the committed step. So in step 4, fructose 1,6-bisphosphate is cleaved by fructose 1,6-bisphosphate aldolase. The names of the enzymes aren't super important. It's just important to sort of follow the general structure of what the molecules, what's happening to the molecules in each step. So in this step, we're turning them into 2 3-carbon sugars. So we're finally breaking those 6 carbons apart. And we are left with glyceraldehyde 3-phosphate, which is also known as GAP, and dihydroxyacetone phosphate, or DHAP. So this step is reversible again, and the remainder of the glycolytic pathway will oxidize 3-carbon sugars. So GAP can continue to step 6, but DHAP will undergo a conversion in step 5. So basically, it's just an isomerization. They have the same atoms, DHAP and GAP, but we just need to convert DHAP using triose phosphate isomerase. So after steps 4 and 5, one glucose molecule has been broken down into 2 molecules of GAP. Although at equilibrium, the mix is 96% DHAP, the use of GAP in subsequent steps pulls the reaction forward. So DHAP and GAP are going to be in equilibrium with each other, but because we can't use DHAP, the only usable form of the product is that GAP product. It's going to be pulled into other steps. And because we're lowering that GAP concentration, it's actually going to pull the equilibrium over from DHAP more so to the GAP. So step 6 is the beginning of the payoff phase. So in this step, we have an enzyme called G3P hydrogenase, and that catalyzes the oxidation of G3P to 1,3-bisphosphoglycerate. This is also a reversible reaction. And this is the first step in glycolysis that actually produces a high energy electron carrier, which is NADH. And that's one of those electron carriers that's going to be really useful for the electron transport chain once we head over. So in step 7, the high energy phosphate on the 1 carbon of 1,3-BPG is transferred to ADP to create ATP via phosphoglycerate kinase. So a lot of big words in that one too, but you just have to understand that in step 7, we are creating ATP. So we're taking one of those phosphate groups from 1,3-BPG and we're putting it back onto ADP to create ATP. And we use the help of a kinase, obviously, to do that. So this is going to yield 1 ATP, and then we're left with a molecule called 3-phosphoglycerate, or 3PG. So step 8, 3-phosphoglycerate is converted into 2-phosphoglycerate by the enzyme phosphoglycerate mutase. So the phosphoryl group on carbon 3 is removed, and a phosphoryl group is added to carbon 2, hence the name of the product molecule. This step is also reversible. So again, this is sort of just an isomerization. We're moving the phosphate group from the 3-carbon to the 2-carbon, and it is reversible. So step 9, right after, 2-phosphoglycerate is dehydrated into phosphoenol pyruvate, and it's catalyzed by an enzyme called enolase, which is also a reversible reaction, but PEP can now enter the final step of glycolysis. So we finally have that PEP, phosphoenol pyruvate, which is a pretty important molecule because we can get over to step 10. And in step 10, pyruvate kinase is what catalyzes the conversion of PEP into pyruvate by transferring, its phosphoryl group to ADP to create ATP. So we're just getting rid of that phosphate group on pyruvate. We're transferring it to an ATP. And in doing that, we create pyruvate and an ATP molecule. So the product pyruvate is created in its enol form and quickly tautomerizes to the keto form. So this tautomerization pulls the entire reaction forward and renders step 10 irreversible once pyruvate is formed. So this is not an equilibrium reaction. It's going to be completely pulled over to pyruvate. So the 2 pyruvate molecules formed by glycolysis will head to the pyruvate dehydrogenase complex to be converted into acetyl-CoA before the Krebs cycle. So we could take a look at that also really quickly because that's a pretty important step before we head over to the Krebs cycle, because you need that molecule acetyl-CoA in order to spin the cycle. So there's a process that we need to bridge the gap from pyruvate to acetyl-CoA. So the complex is actually made up of 3 different enzymes, what we refer to as the pyruvate dehydrogenase complex. So there's E1, E2, and E3. So E1 is also known as pyruvate dehydrogenase, E2 is dihydrolipoyl transacetylase, and E3 is dihydrolipoyl dehydrogenase. So after one glucose molecule makes it through the glycolytic pathway, 2 pyruvate molecules head to the mitochondrial matrix to the pyruvate dehydrogenase complex. So remember, we've been in the cytoplasm for all of glycolysis, and once it heads to the pyruvate dehydrogenase complex, it starts heading over to the mitochondrial matrix. So in the PDC, pyruvate undergoes oxidative decarboxylation. So remember what decarboxylation is. It's that release of carbon dioxide. So a carbon is released from the 3-carbon sugar as CO2, and the remaining 2-carbon entity is attached to a CoA group to form acetyl-CoA, because that remaining 2-carbon entity is an acetyl group. So we attach that to CoA, and that's how we get our molecule acetyl-CoA. So a molecule of NAD plus is also reduced to NADH. So we're in this process, we're also making another NADH, another electron carrier. And PDC is crucially regulated by AMP levels. So AMP levels indicate low energy in the cell and therefore upregulate PDC activity to feed the Krebs cycle. So AMP is what happens when you take 2 phosphate groups away from ATP. It's adenosine monophosphate. So if there are high AMP levels, that usually correlates with low ATP levels. So the cell can use AMP levels to actually detect the level of energy in the cell. So if those AMP levels are low, we're going to upregulate PDC and we're going to spin the Krebs cycle. Okay. So this is the first review question. Take a minute, read this, pause the video. I would take no more than a minute. After you unpause, we're just going to go over it really quickly. Okay, at various points in glycolysis, NAD plus is used to oxidize different molecules. Which answer best describes what would happen if the body supply of NAD plus was allowed to decrease? So the correct answer is D. And let's go over it. So A says, since the main product of glycolysis is pyruvate, its productivity is not affected by NAD plus levels. So that is not true, because remember, we need NAD plus in glycolysis because it acts as that oxidizing agent. So it's taking those electrons from some of the compounds that we needed to take electrons from. So if we don't have that NAD plus, glycolysis is going to be inhibited. We're not going to be able to reach those final steps because we have nowhere to bring those electrons. So A is not correct. B states a decrease in the NAD plus levels would speed up glycolysis as it tries to replenish the levels back to normal, which again is not true. Decrease in the NAD plus levels would severely hinder glycolysis, and glycolysis doesn't replenish NAD plus levels; it replenishes NADH levels, which is the reduced form of NAD plus. So B is also incorrect. C, glycolysis would slow down because there are no longer enough NADH molecules to act as a reducing agent. That's not true. NADH is a reducing agent, but glycolysis wouldn't slow down because there's no longer enough NADH. The reason why it's slowing down is because which is D, is because there are no longer enough NAD plus molecules to act as an oxidizing agent. So if we have nothing to oxidize, then we're not really doing anything with the energy, and glycolysis is going to slow down. So D is the correct answer.